afternoon and warm welcome to all the participants of the national webinar on diagnostic tools in biopharma, pharma, and chemical industries. At the outset, wish you all a very good health and a positive attitude as we are collectively facing the pandemic threat that is sweeping across the humanity beyond borders and oceans. In the current situation, with the interpersonal communication reduced to a minimum, the best opportunity to all of us is to share the information through digital platforms. Hence, we are using one such platform to share and spread knowledge and not corona. As we all know, to prevent the spread of any pandemic disease, vaccines and diagnostic tools play a very important role. India produces 60% of the world vaccines. The diagnostic tool can supply the critical data that can help to determine the best choice of the vaccine. Hence, in this webinar, the speakers will emphasize on the use of the diagnostic tools such as real-time polymerase chain reaction test, mass spectroscopy based proteomics protocol, and X-ray powder diffraction to combat the spread of the disease. For organizing this webinar, I would like to thank our college management and principal for being the constant source of encouragement and support. The achievements of Khalsa College increased to leaps and bounds under the leadership of our principal, Dr. Kiran Mangaonkar sir. To name few achievements, our college received Best College Award by the University of Mumbai. It was recognized by Scientific and Industrial Research Organization. Our college was awarded the fifth grant by Department of Science and Technology. It was awarded the Star College status by the Department of Biotechnology, Government of India. And our college also established National Facility for Biopharmaceuticals, which was supported by Department of Science and Technology with a funding of rupees 3.85 crores. And recently, our college received RUSA grant from the Ministry of Human Resources Development, Government of India. These are the few achievements of my college. For this webinar, I would also like to thank the core team comprising of Dr. Adita Rani, Dr. Abhimanyu Yadav, and Dr. Preeti Khetkar for their time and support in arranging this seminar, and the technical team comprising of Mr. Elvin Jolly, Mr. Shyam Mohan, Mr. Ashish Jaiswal for fixing the wrong things and helping to achieve our goals. Last but not the least, I would like to thank all the faculty members of chemistry department for portraying an attitude of cooperation and offering their valuable inputs from time to time. Finally, without you, dear participants, this webinar would have been meaningless. So I thank all of you for your overwhelming response. The participation is from all over India, stretching from Gujarat to Patna and from Delhi to Kerala. So I welcome once again all of you and hope you will have a wonderful session, enlightening session with our speaker. Now I'd like to share the message to all the participants given by our principal, Dr. Kiran Mangaonkar sir.
Good afternoon. I'm happy to welcome you all for this national webinar on diagnostic tools in biopharma, pharma, and chemical industries, which is organized by Department of Chemistry, Guru Nanak Khalsa College, Mumbai. Today, the whole human race is fighting a war against coronavirus, but earlier also we have experienced such dreaded diseases like SARS, Ebola, swine flu, and in the past Spanish flu in 1920, creating a big havoc in many countries. And of course, we can expect such kind of viruses in the future also. Scientists all over the world are always trying to find out solutions for the diagnosis and cure from these pandemics. Pharmacogenomics and personalized medicine have become major buzzwords in public health and healthcare. They relate to technologies such as genomics, proteomics, microarrays, and sophisticated immunoassays, which have the potential to deliver medicine tailored to an individual's biology or specific pathology of their diseases. We can see the ample possibility of growing diagnostic industry in the wake of personalized medicine. In this 3-day national webinar, experts like Dr. Deepak Thakur, Dr. Krishna Khairnar, and Dr. Sachin Bhandare are delivering their talks, mainly covering the instrumental techniques such as real-time PCR, mass spectrometry, and XRPD. Some of them will be talking on their diagnostic tools for COVID-19 patients. I'm sure this webinar will greatly benefit all the participants. I'm thankful to Dr. Meeta Rakesh Madam and all my colleagues from the Department of Chemistry for organizing this webinar. I wish you all the best. Thank you, and over to Dr. Meeta Rakesh Madam.
Thank you, sir, for highlighting the theme of the webinar. It's a great honor to present before you the first speaker of the webinar lecture series, Dr. Deepak Thakur. Dr. Deepak Thakur is an accomplished person with an illustrious career. He is an alumnus of University of Mumbai. After completing his MSc, he joined Barnett Institute in Boston for PhD in analytical chemistry. His doctoral study was mainly on the development of sensitive high-performance analytical methods for the characterization of the samples of clinical and biopharmaceutical importance. After completing his PhD, he joined as analytical head in Novartis Vaccines and Diagnostics in Cambridge. And in the year 2013, Dr. Deepak Thakur returned to India and then joined Wockhardt Biopharmaceutical Research and Development Group. And finally, in the year 2018, he founded his own company called ACRNS Analytical Technologies, which provides consultancy to bring analytical techniques to biopharmaceutical developers. Thank you so much, sir, for accepting our invitation. The key takeaway from his lecture is to learn and understand what is mass spectroscopy based proteomics and how it can be used as a diagnostic tools for the COVID-19 patients. We thank you once again, sir, and your expertise is of great value to our webinar. Over to you, to the first speaker, Dr. Deepak Thakur. Sir, you can. We cannot hear you, sir. Hello? Sir, you have to unmute. We cannot hear you, sir.
Can you hear me?
Yeah, right. Yes, yes, sir. Yeah. Welcome, sir.
Thank you. Can you see my screen?
No, we cannot, sir. Present now and then, I think you have to go and click on the share.
Yes. hope
Once you
you,
click on that,
yeah,
yeah.
yeah. Okay. So thank you very much to Dr. Meeta, Rakesh, and Dr. Mangaonkar for this invitation and for this opportunity to present a topic, mass spectrometry-based proteomic protocols as a diagnostic tool for COVID-19 patient. This is a very good initiative, as we know we are in a situation last 2-3 months where all our activities are at standstill, but the best use we can make of this time is to understand that in the past there is a lot of literature and research happened on this topic, and were we ready for such a pandemic? So we were not ready. Suddenly there was a rush to find a cure for this disease, a vaccine for this particular virus. So in this presentation we'll look at what is the history, what type of viruses were already identified, and what is the difference in the new virus, and what are the drugs which are targeting which type of proteins. And further, we'll look at diagnostic tools, the diagnostic tools which are already available, and the diagnostic tool using mass spectrometry based, how that is going to add value to overall tools we have for diagnosis. So thank you very much again, and I'll begin. Myself, Deepak Chopra, I'm a founder of ACRNS Analytical Technologies. This particular company, which I'm running for last 2 years, is into training, analytical consultancy, and distributorship of protein metric software for mass spectrometry data, for which is coming out of of the protein characterization, especially for biopharmaceutical development. So outline of the talk is I'll talk about different type of coronaviruses which are known to us, what are the therapeutic approaches, and how proteomics, mass spectrometry based proteomics can be used as a diagnostic tool. We will discuss 2 case studies which are latest developed, and we'll look at it how mass spectrometry based proteomics tool can be used as a diagnostic tool. So coronaviruses, they are named as coronaviruses because of crown-like spikes, the protein spikes which they have on their spherical surface. Since 1960s, these type of viruses were known, and this is the SARS-CoV-2, which is a novel coronavirus which we are now facing, is the 7th in this list. So what is that which is makes this virus unique? First of all, viral factors. So the virus is spherical in structure. It has proteins on its surface, and it's originating from bat. Further, through domestic or other intermediate host, is coming to human and is spreading human to human. These viruses have S protein on their surface. This S protein, as you see, the pink spike on the surface, that's a S protein. It has 2 subunits. S1 is the 1 unit which is responsible for binding with the receptor on the host cell. And S2 is responsible for fusion of this virus with that of the host cell. So host cell is the cell where they are invading. For example, if they invade in humans, our lung cells are the host cells. So S proteins are responsible for attaching these viruses to the host. Further, there is M protein, which is responsible for transmembrane transport of nutrients inside this particular virus. There are other type of proteins too in the overall composition of this virus. Very important is N protein, which is actually making a shell next to the RNA, which is a nuclear material, and which is responsible for multiplication of this virus in the host cell. So these are the M and the N proteins, and further the host cell, that is human, suppose the humans are infected, then human cells which have angiotensin converting enzyme as a receptor. So this the S protein binds to this particular receptor and enters or transfers its nuclear material inside the host. Who are susceptible to this disease? Elderly people above 65 age, those people who have already hypertension, diabetes, chronic pulmonary disease, and cardiovascular diseases. And what are the critical conditions which are we see when patients get infected due to COVID-19 virus? Cytopathic, that means cells are diseased. Then cytokine storm. Further, hypoxia, that means the oxygen level is reduced. Septic shock, that means there is an infection. And what's happening next is the severe complications, that is difficulty in breathing, respiratory distress syndrome. There is a infection which enters into the bloodstream and reaches to different organs. So multiple organ failure. So this is what the mechanism is which results into disease state. And the understanding the genetic material which is present. So the genetic material which is present in the COVID-19 virus is plus single-stranded RNA DNA. Stranded RNA. Further, this particular slide shows us that how this virus binds to the surface angiotensin converting enzyme is a receptor. So enzyme receptor to this, it binds and it releases the RNA material, which is a genome of this virus. It releases that material inside the cell in the cytoplasm and in the cytocell. And then what happens is there is a translation of this particular RNA and generating proteins. Further, subgenomic RNA transcription happens. These subgenomic RNAs generate or synthesize proteins which are responsible for overall new virus formation. So what we see here is the binding, then further proteins translation to generate polyprotein, further subgenomic RNA transcription converting to protein, and the new viruses are generated. That means multiplication of virus happens. So important stages is surface spike protein binds to the receptor. Second is virus nuclear material enters inside. There is a translation, formation of the assembly, and this assembly matures as a new virus. So what are the theoretical, what are the potential therapeutic approaches? You can block the spike protein. You can block the another surface protein which is called as TMPRSS2, which is also responsible for fusion of virus with that of the host cell. You can bring in a soluble receptor so that receptor will bind to virus and will not allow virus to bind to the receptor which is present on our cell. So like this, we'll see on in the next slide, there are different ways we can stop this virus from multiplying. And one of the drug which is called as remdesivir is targeting RNA-dependent RNA polymerase. So this RNA polymerase protein is targeted using this particular drug, and which prevents that polymerization, that RNA-dependent RNA polymerase, which is responsible for replication of viral genome, which is a RNA. Like this, different proteins, enzymes which are involved in this particular the replication process can be targeted using these drug candidates. There are 160 candidates under study, under trials, which are being targeting different components of this multiplication process. And why we are discussing those in this particular presentation is these proteins, if identified during our diagnostic process, gives us, because these are the unique proteins belong to this virus, or some of them belongs to virus and some of them are involved in this replication process, will give us an idea that which one to target as a drug target and which one to use as a biomarker, as an indicator of presence of virus in our body. So we'll look at existing diagnostic tools. The first tool is, which is widely used, is called as reverse transcriptase polymerase chain reaction. What happens here is the DNA of virus is converted to the cDNA using reverse transcriptase. And further, this DNA is multiplied using polymerase chain reaction. In this particular process, fluorescent dye gets bound to every copy of DNA which is generated. And as the multiplication happens, so if the sample has a infection, has that particular RNA, then there will be a replication of DNA and you will see a signal increasing. The other way of detecting or diagnosing this particular disease is detecting the antibody which are generated due to the infection or the proteins which are bound to the virus. So those proteins which are generating the antibody response, they can be detected in the blood, and that test is using enzyme link, that is ELISA test, enzyme link immunosorbent assay, where the antigen which we know the antigen which causes antibody response, we use that antigen which is on the plate and we bring in the blood sample. The antibody will be bound to it, and then there is a secondary antibody which binds to the primary antibody, and secondary antibody has a enzyme which, when comes in contact with the substrate, changes the color of that substrate, and that's how the readout happens. These are the two techniques. RT-PCR is widely used for identifying the RNA of virus, whereas the ELISA-based technique is used for detection of antibody response, the immune response to the presence of antigen. What we'll be talking about, and I'll be presenting the work of the two labs, which are going to use mass spectrometry-based proteomics for identification of the changes in the proteome which are taking place. And this proteome, mass spectrometry-based proteomic methods or protocols will be used for identification of those proteins which are indicator, which are biomarker of presence of this disease. So what is proteome? Proteome is a set of proteins expressed in the cell under given conditions. So disease proteins will have different type of proteins, and or may have same proteins, but their abundances will be different. So study of this proteomes, collection of proteins, thousands of proteins are present in the cells. They have different functions. So study of these proteins is called as proteomics. So proteomics is used to investigate protein expression, rate of protein production, protein degradation, then post-translation modifications which are present on it. What is post-translation modification? One of the example is glycosylation on proteins. So glycosylations are very important. They are indicator of different changes which are happening in the cells. Then movement of proteins between subcellular compartments. Then involvement of proteins in metabolic pathways. Some of the metabolic pathways are overexpressed, overregulated. Some are so upregulated and downregulated, and the interaction between these proteins. All these things can be studied using the proteomics. So to investigate proteome in depth, several high throughput technologies have been developed and most commonly applied are mass spectrometry based proteomics approach, where tandem mass spectrometry is used to identify proteins, not only measure their masses of peptides. The proteins are identified by identifying identifying peptides. So I'll talk about that approach. So here mass spectrometry is primarily used for identification of proteins and their up and down regulation. So we'll talk now about mass spectrometry. Liquid chromatography connected to mass spectrometry is widely used to study proteins, their expressions, their identifications, their differentiation, their up and down regulation between two types of samples. So HPLC is the front end tool, which is or instrument which is used for separating the complex mixture. And further, this complex mixture is sprayed in the mass spec. So the role of mass spectrometry is to measure the analytes. And what is required for measurement of mass of these analytes, that they need to be charged. So what is measured is mass to charge ratio. Proteins are converted through enzymatic digestion into peptides. These peptides are separated on chromatography. They are introduced into the mass spectrometry through ion sources. So what is the role of ion source? Ion source provides charge to the analyte. They provide charge to the analyte. They make these analytes charged so that they can be measured in the mass spectrometry. So the different components we see here of mass spectrometry is HPLC, then ion sources, which are responsible for ionization. Further, the different components of mass spectrometry are different type of mass analyzers. There can be one mass analyzer just to measure the mass, but if we want to measure the mass and further want to fragment that mass into its fragments in order to understand sequence of amino acid which is present in the peptide. So amino acids are present in the which are the building blocks of peptides. They are sequenced in a particular manner, in a particular sequence they are positioned. That sequence is important for identification of protein. So further, the next slide shows us that electron impact ionization is one of the ionization technique. What happens here is, if your molecule is volatile, then you will be separating those molecules by gas chromatography, and those will be subjected to a electron impact ionization. So what happens in electron impact? Very highly energetic electrons are bombarded on these analytes and ions are generated. When we use liquid chromatography for separating compounds, then we use electrospray ionization. By name, electrospray means the application of voltage to that of the metal tip through which eluent is coming out. When voltage is applied, the droplets are generated, and from these droplets, solvents evaporate. As solvent evaporate, the molecules which are present in the droplets, they are of the same charge, and there is a repulsion. There is a huge repulsion between the charged ions. There is a Coulombic explosion which causes these ions to separate, and now the ions enter into the mass spec for their measurement. So we'll look at some other type of ionization sources also, like atmospheric pressure chemical ionization. Here, a corona discharge is used to transfer a charge from a solvent molecule to the molecule of interest. So what is happening here is that if the molecule can take a charge, you can use electrospray ionization. If molecule cannot take charge on its own, but you need to support it by transfer of ion from solvent to the molecule of interest. Then you use atmospheric pressure chemical ionization. Another technique where you can directly generate ions from solid molecule is multi ionization technique. So as you see on this particular slide, on left hand side bottom part, you will see that there is a target plate, and on this target plate your analyte is spotted. Now your analyte is in the solid state, and with them you have matrix compound. What is matrix compound? Matrix compound is an acidic compound, which is when this both the molecules or both the molecules and compounds, when using the laser, when they are dissolved, when that means they are brought into the environment using the laser, then in the gaseous state, the matrix molecule is providing a proton to your molecule of interest. And now through this transfer, your molecule is in the ionic state, and now it can be measured by the mass analyzer. So there are more than 7 to 8 different type of ionization methods. The two which are relevant to us right now, one is electrospray ionization and second is multi-ionization. So once we ionize molecules, the next step is to take them and measure this mass. But the masses will not be measured as masses. Masses will be measured as mass to charge ratio. That means these molecules, analytes, which are converted to ions, they can have single charge, they can have multiple charges. So if they have multiple charges, then mass to charge ratio. If charge is higher, mass to charge ratio will be lower. And what is measured in the mass analyzer is mass to charge ratio. And by determining what is the charge present on that particular analyte, you can calculate what is the mass present. I'll repeat, what mass analyzers measure is mass to charge ratio. And once you determine what is the charge on it, so multiplication of mass to charge ratio with that of charge tells you what is the mass of that analyte. So there are different principles based on which mass to charge is measured, and we'll go over them because this gives us an idea that which type of mass analyzer should be used in which cases. So the very first one, mass analyzer, which was invented was quadrupole mass analyzer. By name, it's a quadrupole. That means 4 poles, 4 rods. These 4 rods have space in between, and they are placed equidistant from the central axis. And voltage is applied to them. A alternating voltage and direct voltage is applied to these 4 rods. And what happens here is the alternating voltage changes the polarity on these quadrupoles. The opposite poles have same charge, and therefore the molecule which tries to enter inside this quadrupole is continuously attracted and repelled. So continuously the molecule is moving in a wave-like form, in a wave-like motion, and if its trajectory, that means the trajectory of a particular ion which is having a mass to charge ratio of a particular number, it will pass through this quadrupole only when it matches a particular frequency. So the application of DC voltage and AC voltage, it is ramped. Ratio is kept constant, but their values are ramped. And as these values are ramped, different M by Z values, the analytes with different M by Z values are allowed to pass through this quadrupole and reaches the detector, and it gets detected. So the masses are measured by scanning the DC and AC voltages. Different ions at a time, one at a time, are allowed to pass and are measured. So what is the plot we get out of it is mass to charge ratio versus intensity. So here the principle was one ion is stable at a particular combination of DC to AC voltage. The other principle based on which ions are separated is when these ions are subjected to a magnetic field, their path is deflected. When the M to mass to charge ratio is high, that means the molecule is heavier, the deflection is lower. But when the molecule is lighter, deflection is more. So based on where it is getting, to what extent it is getting deflected, gives us an idea what is its mass to charge ratio. Further, when the ions are made to fly in a time of flight tube, where at the bottom it has a reflectron. So it is traveling a U-shaped path, and when it travels a U-shaped path, what is happening is the molecules which are lighter will move fast, the molecule which are heavier will travel slowly, and based on the time required for an ion to travel, their mass to charge is determined. Recently, Orbitrap is a mass analyzer which is having a high resolution. And why it has a high resolution? Because the ions are made to move, orbit around a central axis of this particular mass analyzer. And based on the frequency of its movement, the frequency with which it is moving around this axis, that frequency is converted into mass to charge ratio. So Orbitrap is a low resolution mass analyzer, whereas Orbitrap is a high resolution mass analyzer. So what we have seen two major components. One is the ion source, and then inside, when the ions are introduced in the mass pair, these mass analyzers, multiple mass analyzers are connected to each other to perform multiple activities. For example, the first mass analyzer will be responsible for only measuring the precursor masses or the masses of the components we see or we are separating on the chromatograph. Further, when these masses are measured, they need to be fragmented to understand their composition. So another mass analyzer is used to measure these fragments. And in this way, multiple different type of mass analyzers can be combined together to perform mass measurement and fragment measurement. So this is the information about what happens in the mass analyzers. So I'll just now little bit diversion towards the protein structure, and I'll tell you why it is important to know that protein has primary structure, protein has secondary structure, protein has tertiary structure, and protein has quaternary structure. When we study proteins, as we are looking at proteome using mass spectrometry, it is important to know that the impact of proper native structure of protein on its binding to that of its receptor. Also, when we look at protein and when we want to find out identify a particular protein, we need to process the sample in the right way. So protein is a polymer, is made up of amino acids, and amino acid, as name suggests, is a molecule which has amino group and acid. Two amino acids are connected to each other by a peptide bond. As you see in the figure, in this particular picture, you see that there is a peptide bond which is connecting two amino acids. This way, the amino acids are connected to each other, and there is a primary chain of amino acids, which is a primary structure. But these amino acids, this particular primary chain, takes different type of structural motif. One is alpha helix, beta sheets, beta turns. So these are the secondary structural motifs which are dependent on what is the nature of amino acids. What are the side chains present on amino acids? So when you look at left-hand side, lower part of this slide, you will see that the different amino acids are due to the side chains, different side chains, and due to the nature of side chain, these amino acids have properties such as they are aliphatic in nature, they are aromatic in nature, they are polar in nature, they are non-polar in nature, they are charged, they are non-charged, they are acidic, they are basic. So the side chains of these amino acids are responsible for that particular property of the amino acid, and that those properties are responsible for formation of alpha helix, for formation of beta sheets. Non-formation of alpha helix, that means there will be some portion in the structure which is not having, that means it's linear in nature. It doesn't form alpha helix. And these are the different motifs which are present in the tertiary structure. So the protein is, and the forces behind alpha helix and beta sheets are hydrogen bonds. The hydrogen bonds are the bond between heteronuclear atoms like nitrogen and oxygen. So the NH group, the proton which is involved in the NH group, is when it comes in a close proximity of oxygen, it forms a bond, and see that bond is strong enough to generate alpha helix, beta sheets, these type of structural motif. These structural motifs together forms tertiary structure, and these tertiary structure, when they come together, they form quaternary structure. As we have seen that the S protein, which is present on virus, has two subdomains, S1 and S2, and S1 is responsible for formation of or binding of the virus with that of the receptor on the host cell. Now, when we want to identify a protein, we want to process this sample, we want to convert this protein into peptides so that we can sequence the amino acid. Because if you have such a huge protein and thousands of amino acids present in it, the sequencing work becomes very difficult. There are ways to sequence, but the easiest way is to convert such a big protein into its small pieces, which are called as peptides. So what is the importance of knowing this particular information about primary, secondary, tertiary, and quaternary structure is, one thing is their quaternary structure is responsible for their activity, and these structures need to be broken down, they need to be linearized so that we can convert this particular protein into peptides, and then we can sequence those peptides. And based on unique sequences in this identification of these unique peptides which are present in a protein, you can identify a protein. So the protocol for converting proteins into peptides and further identification is as follows, and as we see here. Here, the protein sample, suppose we have cells, we have tissue, we have blood samples. It's a very complex mixture of proteins, and we want to simplify it. How to simplify it? We can do fractionation at subcellular level. We can separate proteins based on their molecular weight. We can run them on gel. So if you have these samples, you can run them on gel. You can fractionate them. Then subfractions you can take if the mixture is complex. You can take subfractions of these proteins and you denature them. So by denaturation, we cause these hydrogen bonds to break, and the secondary structure, the tertiary structure opens up. When there are other types of bonds also which are covalent, that is disulfide bonds, which is between cysteine amino acids. These are covalent bonds, To break these covalent bonds, you need to reduce these proteins using reducing agents. And to prevent them from again forming, we alkylate these sulfhydryl groups. That means SH group is alkylated, and this alkylation prevents again the disulfide bond formation. This particular sample preparation step helps us linearize protein, converting it from its quaternary structure, tertiary structure, from its secondary structure to primary structure. Now, protein is linearized, and now we can convert this protein into peptides. But one important thing which we need to do, we need to select a particular enzyme to convert this protein into peptides. And why a very specific enzyme? Because we want that conversion of that protein into peptides in a specific way. The cleavage should happen at a particular defined amino acid. And one of the enzyme, which is called as trypsin, which cleaves at C terminal of lysine and arginine. So lysine and arginine are known to be a basic amino acids because of they have amino groups on their side chains, and their side chains are long. So this enzyme, the beauty of this enzyme, it recognizes specifically these amino acids, and they cleave peptide bond C terminal of these two amino acids. So now the protein which is generated by trypsin digestion has the peptides which are generated from these proteins by digestion of trypsin has lysine and arginine at their C terminal. So see, all the peptides which are generated by this particular enzymatic digestion has lysine and arginine on their C terminal. This also helps you that yes, this particular digestion is that has happened due to trypsin. If there is any other amino acid that shows you that digestion is not due to trypsin, there is any other trypsin, or the sample is digested due to some other enzyme. So having generated these peptides, which are usually of most of these peptides which are generated from many of the proteins, thousands of proteins which are present in the cells, from them tens of thousands of peptides are generated. And then you need a very high resolving chromatography which can separate these peptides, and then the separated peptides, as the complexity is reduced, these peptides are introduced in the mass spectrometer. So when these peptides are introduced in the mass spectrometer, they are introduced through ion sources. That means these peptides which are present in solution state, they are converted into ions, they are converted into gas phase, and then the masses of these ions, these peptides, are measured in the mass analyzer based on the principle of the mass analyzer. As we have seen, the principles are in the magnetic field, they are deflected from their path. In the time of flight, they are measured based on their mass to charge ratio. Heavier one will move slowly, the lighter ones will move fast, and in this way, the mass analyzer separates these ions based on their mass to charge ratio. Having measured their mass to charge ratio, next thing is to fragment them, to understand their fragments. So the next slide will see how masses are measured and how the fragments are measured and how a peptide is sequenced. So what you see on this particular slide is a chromatogram on the left hand side, and many peaks are eluting from the chromatography. These peaks, which are in these components or these analytes, are in liquid phase. They are converted in the gas phase using ion source, and using the mass analyzer, their masses are measured. When their masses are measured, it is called as MS1. What we see are their masses. What is its mass to charge ratio? Further, these mass to charge ratio or these particular charge species are fragmented using collision induced dissociation. So what happens here is these ions are made to colloid with the noble gases, and on collision the molecule breaks down in a defined way. There is a defined procedure, fragmentation procedure, by which the analyte fragments into fragments, and then after measuring these fragments, as you see on the right hand side bottom picture, these are the masses which are measured as mass to charge ratio. These are the fragments, and these fragments, the fragmentation process is very defined. The fragmentation process is very defined. The ions, the fragments which are generated using defined processes, and you find these fragments, and using algorithm, the fragments are used to generate the sequence of amino acid which is present in that particular protein. This sequence is matched with our database. So whole big proteomic database against which these peptide sequences are matched. And based on their matching with that protein which has this sequence, we will identify that protein. We'll say that that particular protein is present in our sample. So this is the peptide mapping process, right from sample preparation to mass spectrometric measurement of mass to charge ratio and its fragments. We identify peptide, and based on identification of peptide, its sequence, we identify the protein which is present in our sample. This way, thousands of proteins, peptides, and proteins are identified during the proteomics approach. And now we'll see that there are two strategies. Using mass spectrometry, there are two strategies by which first thing is discovery is performed, that is called as discovery workflow, where disease sample and healthy patient sample, these two samples are compared. They are taken through this procedure of protein separation, protein digestion, conversion of proteins to peptides. Then peptides are separated on chromatography. These peptides are subjected to mass spectrometry for measuring their mass to charge, and then their fragmentation to measure the sequence of amino acids in the peptide. When we compare two types of samples, that means a sample which is coming from healthy person and the sample which is coming from a diseased person, when these are compared, what you see is some of the proteins which are upregulated, because the pathways which are involved in the disease are responsible for upregulation of those proteins which are involved in the particular function which is upregulated. So when a function is upregulated, the proteins involved in that pathway or that particular pathway, they are upregulated. And the pathways which are downregulated, proteins involved in that particular pathway are downregulated. And what we see is during the comparison that particular proteins are upregulated, particular proteins are downregulated. This particular exercise, which is called as discovery, discovers biomarkers. The biomarkers, if these biomarkers are found in the blood samples, then we can say the person is having that particular disease. Once identifying that who are the biomarkers who are involved in these pathways, which are either upregulated or downregulated, we can focus our mass spectrometric analysis only on those proteins. That means we will not try to identify other proteins. We will look for only those peptides which we know, if present, they are indicator of a disease. So then once we do the discovery, we identify biomarker, the next step is targeted quantitation, where we will be only focusing on those peptides which we know which are the biomarkers. And on the left hand side, when you see, that's a workflow for targeted proteomics, through which we will be looking for only those peptides which are biomarkers. So what happens here is that the similar procedure, sample preparation will be, you extract proteins, you separate proteins, you digest protein, but when you use mass spectrometer, you will be using a mass spectrometer which has more sensitivity and which can focus on one particular mass at a time. And I'll show you in the next slide how this is done, how targeted proteomics is done. So what we see here is the first mass analyzer, which is a quadrupole, selects only the peptide of our interest. So we are asking our mass analyzer to focus on only that mass of the peptide, which is a biomarker. Then this, once that is present, it will be taken to the next quadrupole where fragmentation happens. And after fragmentation, the peptides, the fragments will be generated, and unique fragments will be measured in the third quadrupole, which is again a mass analyzer. So this is a diagnostic tool that we have identified. For example, you have identified that the proteins which are present in the virus, they enter into our bloodstream. And you want to identify what are the proteins which can enter in the bloodstream from the virus. Once you know that, once you have identified those using the global proteomics, using the discovery proteomics, then once you have done this exercise, then you will be doing targeted proteomics as a diagnostic tool. That means you will be taking blood samples of healthy people, those who are suspicious, who are maybe are under suspicion, or may have this particular disease, or those who are confirmed. All these samples, you can take them through proteomic workflow. You process these samples, generate peptides, and then you monitor these peptides using targeted proteomics. The healthy patient or the healthy person will not have these peptides, which are coming from either from pathways which are upregulated or proteins which are present in the virus. So targeted proteomics gives you a diagnostic tool to look for only those peptides which are known to be coming from viral proteins or coming from pathways which are upregulated. Now, having seen or having understood the discovery proteomics and target proteomics, we'll look at the first case study, which is where mass spectrometry based proteomics is used for identification of, or as a diagnostic tool. The proteomics approach is used as a diagnostic tool for some patients. So here, the patients which were already identified using RT-PCR were given a 20 ml of saline solution to gargle. And when this 20 ml of gargle solution was given to them and they gargled and that solution was collected, now naturally that solution will have, gargle solution will have proteins either coming from virus and also definitely the proteins coming from our saliva. So these proteins were precipitated using acetone treatment. So the acetone, which is at minus 20 degrees centigrade, is added. 1 ml of acetone was added to 750 ml of this gargle solution, and solution was stored at minus 20 degrees overnight. This was to precipitate all the proteins from the solution. Then the solutions were centrifuged, supernatant were discarded, and the pellet was now purified from other components, and now pellet is solubilized. One thing which is known is that these viral proteins are glycosylated. That means they have glycans. Glycans are polysaccharides. So the sugar molecules, monosaccharides, they are arranged in a particular fashion. They are covalently linked to each other in a particular fashion. And these glycans, which are made up of different type of monosaccharides, they are attached to protein. And that's why proteins are called as glycoprotein. So if we want to digest this protein into peptides, we need to first remove these glycans. And there is an enzyme called as PNGSF. This enzyme is known to remove N-glycosylated glycans from the protein. So this is the step which is required to remove these glycans, because these glycans which are present on viruses are heavily glycosylated, and the reason for this glycosylation is to evade other proteolysis. The proteolysis due to the proteases. So proteases are those enzymes which cleave protein into peptides, and these proteins which are present on virus, they are heavily glycosylated so that their backbone, protein backbone, can be protected from these proteases. So when we study these samples, we want our protein to be converted into peptide, and therefore first we remove these glycosylations using these enzymes. Once the protein is free from these glycans, the protein is subjected to reduction and alkylation. That means we open up the protein, we want to linearize this protein so that protein can be digested using the trypsin into peptides in a more efficient fashion, because we know our sample is very dilute. As our sample is dilute, we have very low number of proteins, and therefore we want to have an efficient process of sample preparation. So in presence of this buffer, trypsin is introduced in the reduced and alkylated sample, and further the peptides were generated by incubating these proteins which are reduced and alkylated with trypsin for 30 minutes at 70 degrees. One important point in this particular case study and the sample preparation is that this trypsin is immobilized, and otherwise if you use 70 degrees, the trypsin may undergo denaturation and there will be no impact. But these trypsin molecules are immobilized on beads, and by immobilization there is a limitation to denaturation of trypsin. There is a resistance to denaturation of trypsin at higher temperature. Because they are immobilized, we can use them at higher temperature. They are still active. They can act on this our protein of interest, and they can convert them into peptides. So as the proteins are converted into peptides, we do liquid chromatography. Further, if you see here, nano HPLC is used. That means the flow rate is in the nano flow. And why it is in the nano flow? Because as we reduce the flow, the efficiency of ionization increases. Because droplets are smaller, solvent can evaporate faster from the droplets, ions will be formed more efficiently. As ions will be formed more efficiently, there will be more number of ions. The efficiency of ion generation is higher. The chances of detecting and further identifying that peptide is higher. As you see, Orbitrap mass spec is used in this particular analysis, which is targeted analysis. And as I explained to you, Orbitrap is the latest mass analyzer, which is very high resolution. That means accurate mass can be obtained using this mass analyzer, and therefore our identification in this particular exercise where we are doing diagnosis, we want a very confident identification. So we want accurate mass measurement. So a mass analyzer, which is Orbitrap, which is a very accurate mass analyzer, is employed here. Nano HPLC is employed here so that there will be efficient ionization of ions and they will be subjected to mass spectrometry analysis. So this is the application of mass spectrometry for analysis of the gargle samples coming from patients. And now we'll look at what is the result, which type of proteins were identified using this particular platform or a procedure. So we see here on this slide the result, and what we see here is the amino acid sequence of a protein which is present in the virus. This is the overall sequence, and what we see on the top is the A, which is patient A, and the another one is patient B. Now, this particular protein which is identified is called as nucleoprotein. And what is its role? This particular protein forms a shell around the RNA, the nuclear material of virus. When this particular protein was present in our gargle sample, through our procedure, we precipitated that protein, we reduced and alkylated that protein, digested that protein, and if you see the peptides which were found have K on its C terminal. So K stands for lysine. And also some of the peptides which are green in color, if you see the peptides which are fragment of this whole big protein, which is generated using within trypsin, has either K or R on their C terminal. So that means our digestion is specific. We have digested our protein properly and generated peptides which has either K, that is lysine, or R, that is arginine, on its C terminal. So this is a evidence that yes, even at such a low level, protein is extracted, protein is digested, peptides are generated, and those peptides are confidently identified when they were not only measured by the mass spec, but they were fragmented. So let's look at the data which is showing that this particular peptide is measured, not only peptide measured, but even when you see this particular slide where a fragmentation pattern of the peptide which is identified is shown. What you see here in green, if you see in this text, there is a peptide which is written as PGQ, GLP, and so on. These are all amino acids, and this is the sequence. This is around 21 amino acid peptide. When this was fragmented, it fragments between the amino acids and different ions are generated. You see here B and Y ions are written here. So when you see the masses of or mass to charge ratios which are observed here as the fragment, you will find that there are B ions and there are Y ions. So in this fragmentation, the fragments which are generated from N terminal, that means from left hand side, these fragments are called as B ions. Fragments which are generated from right hand side are called as Y ions. So now you see many B ions and Y ions, and based on the masses of these you how these amino acids are sequenced in this peptide. Once the measurement of intact mass and measurement of the sequence, that is this particular process of using this fragmentation data to come to a conclusion, what is the sequence of amino acid in this peptide, we can confidently say that this peptide belongs to the protein, which is nucleoprotein, which is present in the virus. So our proteomics approach identified the protein which is present in the virus. Whereas the current approaches like RT-PCR identifies the nuclear material, that is RNA, which is present in the virus. So the RT-PCR based method which is in use identifies the nuclear material, whereas mass spectrometry based proteomics approach identifies peptides which are unique to the proteins which are present in the virus. We'll also take another example, another case study here, where, so I'll conclude the first study where the samples from these highly diluted gargle samples, a protein was identified which is part of this virus. It's a straightforward method which relies on acetone precipitation, removal of glycan, and digestion of protein using trypsin, followed by targeted mass spectrometric analysis. Using this approach, one can identify peptides which are originating from nucleoproteins, because we identified multiple proteins, not only one protein, but multiple protein which is originating from the same protein. Multiple peptides which are originating from the same protein. And the future plan of this group, this is a lab in Germany, in a school, in a university, where they have demonstrated this particular protocol. They will be trying to understand even the sample which are present in the lungs, because lungs are infected first, and the liquid which is secreting due to multiplication of virus. So if they take sample from there and analyze it, they will get that the liquid they find, whether it's the secretion liquid or it's a liquid which is having viruses in it. So this is the proteomics approach. This is the first case study. What we are going to do is going to look at the next case study, which is another promising study which is going on between a collaboration between the MAP Sciences and CantLab Health Technologies, which are close to delivering a sophisticated and affordable testing technology that will complement the existing technique, which is RT-PCR based. This team is using, again, here they are using MALDI-TOF. So as I explained to you, MALDI is a ionization source, and which is an offline technique. You need not do protein separation. You need not do protein even digestion. You can spot samples on a plate, and you can ionize those samples using matrix assisted laser desorption ionization technique. So laser is used to desorb these molecules. There is a matrix which assists in ionization, and directly the samples which are collected, you can directly introduce them into the time of flight mass spectrometer. So time of flight, we saw, is a mass analyzer which measures the m/z mass to charge ratio, and it is based on the heaviness or lightness of a molecule. So here, combination of MALDI mass analyzer with time of flight mass analyzer. So with with the ionization technique and mass analyzer, this is the mass spectrometer, which can directly measure unique peptides, which are with unique proteins which are present in COVID-19. So this technique will, if you bring the sample within 30 minutes, it will do the sample preparation. That means mixing it with the matrix, then loading it on the plate, performing mass ionization and mass measurement, and further confirm the presence of the proteins which are coming from the COVID. Here, this collaboration is working on analysis of with the clinical team, so with the hospital settings, so that they can procure samples from the patient and they can use this technique for measurement of intact protein. Their claim is that as the sample preparation is easy, operation of multi-TOF mass analyzer is also easy. This can be used as a simple technique to directly measure masses of proteins which are coming from viruses. So let's look at what are the steps involved in that. First thing is you take the sample, apply this sample on multi-plate. So you see here a multi-plate, which is where the samples can be loaded. And at a time, you can load 96 plates, 96 spots, or you can load 396. So there are plates which are bigger, can take at a time 396 samples. So you bring in your samples, spot them with matrix, and load this plate in the ionization chamber and fire the laser. Laser causes desorption, presence of matrix causes ionization of the protein of interest, and then these ions which are generated are subjected to mass analyzer where masses of these proteins are measured. The mass analyzer is time of flight. So based on their flying time, their mass to charge is determined, and in case of MALDI, always the charge is 1. They do not form ions of multiply charge, and therefore the proteins which are present in the sample will be measured, and they will be measured as mass to charge, which is their mass. So as you see in this particular picture, the TOF, the output of TOF is presented as a mass profile. So there can be small molecules, small peptides, or there can be huge big proteins. And TOF has a capability, multi-TOF combination has a capability to measure the masses of intact proteins. So this particular approach can measure directly the proteins coming from virus. You need not do sample preparation. So this is another approach which this particular group has taken, and the efforts are on. This is the initial data. The claim is without processing, you can test, you can use this as a diagnostic tool to analyze 1,000 patient samples in a day. So now diagnosis is important because early diagnosis helps you use proper medicine to prevent multiplication of virus. As we have seen in the initial slide, that the mechanism of multiplication of virus in the host cell can be targeted using the candidates which are in the clinics if we know this in the early stages, or we identify that the patient is infected with the virus. So in summary, what we discussed in this particular presentation is that structural features of coronavirus, that it is made up of different type of proteins. These different proteins have different role to play. They are responsible in binding with the receptor, their entry, their nuclear material entry inside the host cell, then there is a replication, and they can be targeted. So if the targets are known, drug development can be done to target those proteins which are involved in replication process. Then we discussed that how mass spectrometry based proteomics approach can be used for biomarker discovery. That means either you discover those proteins which are involved in different pathways which are either upregulating and downregulating. Those are also indicators, and those can also be targeted for a clinical or as a diagnostic tool. Further, we looked at two approaches where direct protein can be detected and can be used as a diagnostic tool, or the whole mixture of proteins is isolated, glycans are removed, proteins are converted into peptides, and then those unique peptides were measured using targeted proteomics to confirm that yes, this particular patient is infected, or this particular patient's throat sample or gargle sample has virus. So this is the early detection. You come to know in early stages that there is a infection to a particular human being. So this is a summary of what we discussed today. And further, I want to acknowledge Dr. Meeta Rakesh for this particular opportunity. And I have interacted with many faculties of Khalsa College. They have attended our programs, and even students. And yes, all the laurels and honors they have received as the best college is evident in that interaction. I have seen that the faculties are taking so much of efforts to educate their students about newer technologies. They themselves are getting aware of these technologies and then passing this information to the students. My colleague in my company ACRNS, Ms. Riti, Ms. Vaishnavi, and Mr. Shriyari, they have helped me with preparation of this presentation, lot of inputs on a lot of technicalities in the presentation, and a lot of knowledge which they have shared with me to make this presentation successful. So I'm thankful to you for your attention, and now forum is open for the question answer session. So we have questions, and I'll read the question and I'll answer them. So what do these techniques use as a sample? So Mrutyunjay has this question that what do these techniques use as a sample? So these techniques can use samples as we saw. First one can be a blood sample. The second one can be a gargle sample, isn't it? So when we do a targeted analysis, we'll be not looking for tissue sample, but we'll be looking for samples like gargle sample, which are not invasive. They are not causing trouble to the patient, just a gargling, or a blood drop, just a blood drop, which is good enough for doing multi-type analysis. And as you mentioned, as I have presented in this presentation, that the viruses which were present in the throat, the gargle sample is actually diluting those samples, but still you could identify the peptide, and attomole level sensitivity is there in the proteomics approach. When you do targeted approach, attomole level, that means 10⁻⁻18 mole, that concentration you can detect for a peptide. Next question is, what about the cost for testing the single sample? So as I mentioned, that the peptide-based method, which is at this moment, is taking around 3 hours time. So whole sample preparation is 1 hour overnight incubation, then peptide generation, and chromatography time is 3 hours. So it's taking almost a whole day. And the cost will be, as you know, you are using a mass spectrometer. These mass spectrometers cost something in the range of 3 crores to 7 to 8 crores. They are very costly machine. But once you do the discovery part, then you can go to triple quadruple, which are easier. And these machines cost around 1 crore or 1.5 crore. So they are not costlier. And then you can do diagnosis on less costlier machine once you know what are the biomarkers. So exact cost calculation I have not done, but as you have more samples and you run it on the machines where you are just doing targeted analysis, the cost will be low. The next question is whether high resolution mass spectrometer can be used to determine exact mass rather than using mass analyzer. Yes. So as I mentioned to you, quadrupole is low mass resolution. And when you talk about Orbitrap or time of flight mass analyzer, they are high resolution mass analyzers. And they are used for exact mass because we don't want to have a false positive or false negative. We want to have a confident identification. High resolution mass spectrometer based work will definitely help in no false positives. Further, the question is from Snehal, how much time required to complete analysis of sample? So as I mentioned to you, there are two approaches, and as the sample preparation approaches are improved, the time can be reduced. But at this time, what I see is a overnight sample preparation, then next day digestion, and then again 3 hours chromatography for one analysis. So almost pretty much 24 hours for at this stage. But if you go for identification of a intact protein, it takes 30 minutes. So we can have complementary techniques to initially qualify a marker, and then we can reduce the time. Then Agnet has a question that how the proteins from samples of patients help in identification of disease. Yes. So, for example, if you first take samples of protein, samples from patients, those are definitely proved. Their RT-PCR based method has also already demonstrated that they are patients. So this actually helps us develop a method, looking for what are those proteins which can be easily observed, because ionization of peptide is, the peptide which can be easily ionized and identified. We have to select that peptide as a marker. So selection of first sample of patients help us what to expect. And then you go for healthy people or those people who are not identified as patients, and then try to find out whether they have those markers or not. Further, Nityunjay has a question that when we talk about proteomics analysis of sample preparation, yes, sample preparation has a major step. But if you use methods where if you improve methods, and there is an opportunity of improving methods doing only targeted analysis. So there are opportunities of developing low cost methods and also which are focused on proteins. See, another benefit is if there is a mutation. Suppose there is a mutation in your RNA, and then there is a protein and the proteins are known, which are getting generated. In mass spec, as you are measuring samples, you will see that the masses are changing. And if you see masses are changing, that means there is a mutation happening, and that's the evidence. Directly you are coming to know that yes, masses is testing of COVID by RT-PCR is 100% accurate. See, what we are looking at, because we know the RNA material and during RT-PCR method, you are using specific primers to measure and multiply these the DNAs, which are, so from you first of all convert this RNA to DNA, and then you multiply DNA. So over there you use specific markers, specific primers. So whole process involves specificity. So they are accurate. There is, there is a work done where they take samples from influenza. That means people who have flu, And they demonstrate that yes, using this RT-PCR, they are not detected. What is detected is the right patient. Further, Akansha says bioinformatics be used to detect the virus in samples to be analyzed. So if you see bioinformatics, application of bioinformatics, whether you speak about um, the interaction between the drug and targets, that sort of work, or the work where you just want to see the pattern. For example, the second approach where multi-MS is used, they are looking for a pattern, not just one protein, but the pattern. So yes, computation is used to look for a pattern and how these patterns are changing when person is just infected, when the person is fully infected, the patterns are changing. So yes, you can use these computational techniques to look at the pattern. Then Bhagya has a question. Immediate analysis of sample to be done or can be stored if there are large number of samples. Yes, if there are large number of samples, we have to follow some precautions. And what are the precautions? There should be the compounds which resist proteolysis of our protein. So we have to preserve our sample from proteolysis. So those precautions needs to be taken. Very good question. Further, Jayachandran has a question. Will this technique pave way for the medicine for COVID-19 as protein is identified? See, proteins are already identified. We have complete information of what type of proteins are there and not. The techniques right now employed are for diagnostic purposes. So proteins are already identified. Proteomics approach has already identified what type of interleukins are overexpressing. So what type of the interleukins are seen as overexpressing. So that data is already available. The medicines 161 latest number, 161 number of molecules are already in trials. Or people are studying them. That how can we target or how can we stop multiplication of virus in the host cell? Then what is the accuracy percentage of this technique? Yes. So usually in proteomics, there are two parts. One is you take, for example, you take a protein, convert it into a peptide, and then you do a fragmentation of it. And the fragmentation which is generated depends upon what is the abundance of your peptide. If it is low abundant, its sequence will have less number of the fragmentation pattern will have less number of fragments, and therefore in that case accuracy will be low because the number of fragments we need to accurately predict a peptide has to be high. So now at this moment at attomole level or so, they have already demonstrated that they are getting good sequence coverage. But even below, if you go below that concentration, definitely there will be an issue with accuracy. Then SPP chemistry. So that sample preparation, I believe that's sample preparation chemistry. Oh, I'm sorry. Somebody with the name SPP. If we destroy the surface of virus protein, that is S1 protein, then it may be unable to stuck on host cell, and it is possible to do this. Absolutely. Many a times, protein denaturation, isn't it? Protein denaturation causes that protein to linearize, and therefore that protein will have difficulty binding to its target. So the options, one option can be you circulate your receptor in your bloodstream. Okay, you circulate your receptor in the bloodstream so that that receptor will target virus protein, surface virus protein, and therefore we will delay that protein binding to its target. So the means by which you can denature it or you can block it, you can use small molecules to block to those areas which are responsible for binding to the host. Then Jasveer has a question: how effective the voltammetry and spectrophotometry is going to be for analysis of such sample? See, the RT-PCR method, mass spectrometry based methods are very specific, and that specificness will not be there in spectrophotometric technique in such a complex sample. For example, you shouldn't get a very specific UV absorption or specific functional groups. I don't think that that's possible in case of proteins. Nadine has a question: why only trypsin? Why not chymotrypsin? Very good question. Chymotrypsin specificity is not that good when you compare it with the trypsin. Chymotrypsin cleaves to next to hydrophobic amino acids, and its specificity is not that great as that of trypsin. Another thing is trypsin-lysed peptides. The peptides which are generated due to trypsin are having a lysine or arginine on their C-terminal, and they are beneficial for its fragmentation. So there are multiple purposes. That's why trypsin is used to generate these type of peptides. And also the presence of lysine and arginine in our proteome, their presence is quite significant. And when you use trypsin, the peptides generated are in the range of 10 to 20 amino acids. So peptides have 10 to 20 amino acids. And based on that, it is much easier to do the proteomics. Prajakta has a question: how can we dispose that sample after analysis? I think that's a great question. Think about if we get a sample, and that sample is a gargle sample already in saline, you have solubilized it. So the antigen is already solubilized, isn't it? And what if you want to do? If you want to get rid of its antigenicity, you add something like urea, which will cause denaturation of those proteins, and which will cause reduction or removal of its immunogenicity. So this is what my feeling is. There can be more ways of disposing of these samples. Next is Anu's question on Anu has a question. So how the sample is discarded? That's what we just discussed. Omkar has a question that when we use proteomic based approach for using mass spectrometry, when to use RT-PCR? Very good question. So at this stage, the claim is proteomics is complementary to RT-PCR. That means RT-PCR is a gold standard. But we know there are two types of enzymes which are used. There are primers used, and this is adding to the cost. The cost is very high. So how can we handle this with a technique which is much less expensive? Hello?
Hello.
Yes, madam.
So you can take this last question now.
Okay. Yes. There are only two questions now. Yes. Yeah. So Gopa has a question: which method takes lesser time? As I mentioned, I would prefer to do it offline technique, which is MALDI-TOF, which is going to find the protein, but just based on protein with accurate mass. If we could do confident identification, that will be better. That if there is a confident identification and multiple proteins which are used as significant signature, then that will be a confident identification, not just based on one protein, but significant signature of proteins. Can S protein be destroyed so that it will be unable to bind and fuse with the host cell? See, whole world is, millions of dollars and billions of dollars are being spent across the globe to destroy this protein, right? And the destroying process is either you block it, right? you block it rather from its binding to that of its receptor. So I do not see that there is a candidate in development which can destroy that particular protein. See, think about it. If you destroy this S protein, you will be destroying our proteins also, which are important for our existence. And that is why that path we cannot take. We cannot destroy that protein, because then our other proteins, which are our very important proteins, our cell surface proteins, receptors, they will also get damaged. We don't want to do that sort of damage. So therefore, we are blocking them. We want to block it from its, specifically using a particular drug which can only bind to S protein and will not damage or will not block other protein functions. So, great questions here. I think there were 20 questions, and the way the questions were asked, I think you had really good understanding about the topic and concern about what type of development should be done so that we can get over this particular problem. Okay, so with that, I thank you all for this opportunity to make this presentation. And now the mic will be with Madam. I'll hand it over to her. And yeah, Madam, please go ahead.
Thank you, sir. Very good evening to all. Can you hear me?
Yes, go ahead.
It's my privilege to propose the formal vote of thanks for day one session of the 3-day-long national webinar organized by the Department of Chemistry, GN Khalsa College, Matunga, Mumbai. At the outset, on behalf of our management, Principal Dr. Kiran Mangaonkar and Department of Chemistry, I thank sincerely our today's speaker, Dr. Deepak Thakur, founder of ACRNS Analytical Technologies Private Limited, for accepting our invitation. Thank you, Dr. Deepak, for sparing your valuable time and enlightening us with the various aspects of diagnostic tools in biopharma, pharma, and chemical industries. I'm sure all the participants would agree with me, it was an intellectual feast for all of us. Dr. Deepak, you connected every aspect of the current prevailing hot topic, coronavirus and COVID-19, by explaining its various types, the structural features, mechanisms, drug development, various therapies, and various diagnostic tools. You also explained about the role of mass spectrometry-based proteomic protocols as a diagnostic tool for COVID-19 patients. And also the most important part is you explained about the protocols for the analysis of samples from those patients who are COVID positive. I'm sure it has ignited a spark in the minds of participants with various opportunities of research related to this topic. Thank you, Dr. Deepak, for this overwhelming, informative session. I thank our college management and our principal for permitting us and cooperating with us to conduct this webinar at a national level. I'll be failing in my duty if I forget to thank our dear participants who have attended this session in a very large number from various parts of this country. Thank you all for being a wonderful audience. There is much more in store for you in the coming 2 days. Before I conclude, would like to inform you all to fill the feedback form and attendance without fail. Link for the same is pasted in the description box as well as the chat box. Please fill it correctly and the certificates will be prepared on the basis of feedback forms only. You will get the certificates only on 3rd day of participation. You will receive the certificates within 5 to 7 days post 23rd May. Meet you all again at 3:15 p.m. tomorrow. Last but not the least, I thank the Almighty for keeping us all with high spirits. Stay healthy and stay safe. Thank you all. Aldrin.